Hello everyone! Today we're going to talk about glycolysis and how it is regulated. So the first thing we should point out is that glycolysis is a redox pathway, oxidation reduction reaction. So we start with glucose and we end up with two pyruvates, and that is an oxidation. And you know you can't have an oxidation without a reduction. So the protons and electrons that glucose loses when it is converted to pyruvate, NAD plus picks those up and you get NADH and H plus. So we want to, before we start, talk about how glycolysis fits in with animal metabolism. So for example, the muscle. So the muscle takes up glucose from the blood. The muscle also stores glycogen, which can be degraded to glucose 6-phosphate, and we'll talk about that in a later section. So whether you start out with glucose or glucose 6-phosphate, it's oxidized to pyruvate in the glycolytic pathway. It is an exergonic pathway, so energy is released in the form of 2 ATPs per 1 glucose. And then in the muscle, the ATP is used to power the muscle. So that's the main role of glycolysis in the muscle, is to break glucose down to pyruvate to obtain ATP to power the muscle. In the liver, things are a little bit different. So when we eat a meal, the glucose that is absorbed from our meal goes from it's absorbed in the small intestine, and then it enters the portal vein, and the portal vein goes right straight to the liver. So that means the liver gets the first crack at the glucose that we eat. So what it does is the liver takes up nearly all of the fructose and galactose from our diet, and maybe about a little more than half of the glucose that we eat. And then the liver passes on the rest of the glucose to circulation. So the glucose that the liver takes up is converted to glycogen, and we'll talk about that later in the chapter too. So any extra glucose goes into glycolysis to provide ATP, and then that helps power glycogen synthesis because that requires energy. But glycolysis also provides intermediates, which can be used to synthesize fatty acids and cholesterol. So the main role of glycolysis in the liver is to provide building blocks for biosynthesis. So you would be in a good place to synthesize new molecules after a meal. So we want to talk about glycolysis. We want to talk about all 10 of these steps. And we want to know which are those enzymes that catalyze reactions that are far from equilibrium, the ones with the really high negative delta G values. Because those will be the reactions that catalyze metabolically irreversible, allosterically regulated enzymes. So the flux of the pathway will be controlled by regulating those enzymes. So if those enzymes are turned off, the pathway is turned off. If those enzymes are turned on, then the pathway is turned on. So we want to know what those enzymes are, and then we want to know who is going to be regulating them. So ATP, ADP, and AMP are good candidates, since glycolysis is an ATP-producing pathway. And ATP and ADP and AMP will be good candidates to let us know the energy status of the cell. Those are good candidates, but also metabolites in the pathway are also good candidates. So in the Where Are We section, we are here. So remember, this is my overview of catabolism slide. So we're going from glucose to glucose to pyruvate. 1 glucose goes to 2 pyruvate, although it doesn't say that here. The pathway is called glycolysis. It takes 10 steps. So we're going to divide these 10 steps into 2 phases. So phase 1 is energy investment. So I did promise you that glycolysis is an ATP producing pathway, but we have to spend 2 ATPs first. So we're going to invest 2 ATPs, and then we're going to produce 4. So the net is 2. So in phase 1, glucose will be isomerized and phosphorylated to fructose 1,6-bisphosphate. And then fructose 1,6-bisphosphate will be split into glyceraldehyde 3-phosphate, which is oxidized to pyruvate. And in phase 2, that's energy recovery, where we produce 4 ATP. So the net yield for the glycolysis pathway is 1 mole of glucose gives us 2 moles of ATP. So let's answer that first question. So if you're listening to this now, you have already gone on to Top Hat and you've already done your pre-lecture Top Hat quiz, so you will already know that when you're looking at the reactions of glycolysis, it's the delta G value. that we're interested in, not the delta G zero prime, because that's the involves reactants and products in their standard states, and that's not what we have in the cell. So we look for the delta G's, and if you look at these three reactions, hexokinase, PFK, and pyruvate kinase, all of those have pretty high negative delta G values. So here's another way to look at the same thing. If we look at glucose as being sort of this downhill pathway, you can see the hexokinase, which is step 1, PFK, which is step 3, and pyruvate kinase, which is the last step, step 10. Those are where the big downhill steps are. And if you can imagine riding a bicycle like I like to do, that if I reversed course, it would be fairly easy for me to turn my bicycle around and go on those flat parts, but it's very difficult to go up those hills. So the steps 1, 3, and 10 are the regulated steps. Those go in that direction and that direction only, because those hills are just too difficult to reverse. So this is another graphic that shows the same thing. So if we look at the standard Gibbs free energy changes, we see that those aren't that helpful. And again, those are under standard states, which you don't see in the cell. But if you look at the bottom, you can see, oh, that's right, I have my pointer here. If you look at the bottom, you look at my highlighter right here. So you can see we have the big downhill steps. That's step 1, and that's step 3, and that's step 10. So those would be the most difficult step to reverse, so we do not reverse them. So we want to talk about how this pathway is controlled. So it's important to notice that the cell is sensitive to ATP levels. So a high concentration of ATP means that the cell is energy rich. Whereas if the concentration of ATP is low, but ADP and AMP are high, that means that the cell is energy poor. So when ATP is used, ADP and AMP are products. And AMP rises when ADP does. There's an adenylate kinase reaction right here. So if the ADP concentration goes up, then we're going to push this reaction in that direction. So that's going to give us AMP. So when we're trying to figure out whether an enzyme is turned on or turned off, we have to think about, is the cell energy rich or is the cell energy poor? And if the cell is energy poor, we're going to want to turn glycolysis on, because that's going to help us produce the ATP that we need. So here is the first step in glycolysis. So step 1 is the hexokinase reaction, and we should notice that the enzyme is hexokinase, and the arrow is only going in one direction and one direction only. So glucose enters the cell, and immediately we need to phosphorylate it. So ATP is the phosphate donor, and we get glucose 6-phosphate. So notice this is an energy investment. We have to spend an ATP to phosphorylate the glucose. So we're going to regulate the pathway here at step 1. And the way that happens is fairly simple. The product, glucose 6-phosphate, is an allosteric inhibitor. So that means that when glucose 6-phosphate binds to the hexokinase, and it doesn't bind at the active site, it binds somewhere else, but it stabilizes the T form. So here's a visual. So when glucose enters the cell, the very first thing that happens is that it is phosphorylated to glucose 6-phosphate, and then it enters glycolysis. So we should notice that glucose enters the cell through glucose transporters. Glucose is neutral, and by phosphorylating the glucose to glucose 6-phosphate, we put a -2 charge on that glucose. So that keeps the glucose 6-phosphate in the cell. So the glucose 6-phosphate would not easily go through those transporters, because the transporters are specific for glucose. And also having a -2 charge on the glucose 6-phosphate means it couldn't easily leave the cell. So it's this to the cell's advantage. So whenever glucose gets inside the cell, you put a phosphate on it, and that will trap it inside the cell. All right, step 2, this is an isomerase. So phosphoglucoisomerase will isomerize glucose 6-phosphate to fructose 6-phosphate. This is a near equilibrium reaction, and it is indicated because the arrow is going in both directions. So we can tell at a glance that this is not going to be a regulated step of the pathway. So our next step, we're at fructose 6-phosphate. We're going to carry out another phosphorylation. So remember, we've got a kinase here. And a kinase does, one of the things that it does is ATP is involved, and we're going to do another phosphorylation. And here, we're going to put the phosphate on the number 1 carbon. So now we have fructose 1,6-bisphosphate. So the bis means 2. So we've got 2 phosphates on our fructose. So bis is different than di. This is adenosine diphosphate. Di also means 2, but if you look at the structure of ADP, the phosphates are next to each other. So if you say bisphosphate, I have 2 phosphates, but they're not next to each other. So we can see a couple things. One is that we are spending our second molecule of ATP, so that's an investment. And we can also see that the artist has drawn this arrow in one direction. So that means that the reaction occurs in only one direction. It's not reversible. So this is a regulated step. So it's not just a regulated step, it is the regulated step. This is the most important step to regulate glycolysis. It's the major control point of glycolysis. So you might have wondered why the major control point wasn't hexokinase, because that's step number 1 after all. So why not regulate the pathway here? Well, here's a little bit of a sneak preview. So glucose is converted to glucose 6-phosphate, and today we're talking about how we're going to take that all the way to pyruvate. But the glucose 6-phosphate can enter the pentose phosphate pathway. It can enter glycogen synthesis. So the phosphofructokinase 1 reaction is regulated because this is the first irreversible reaction that's unique to glycolysis. So once you do the PFK reaction, that pretty much commits you to carrying on. So I have some links to the animated figures, and since I'm using Screencast-O-Matic now, I can capture those for you. So here we have later on, we'll talk about glycogen being broken down to glucose 6-phosphate. And glucose 6-phosphate can be used as a reactant to synthesize glycogen. Glucose is converted to glucose 6-phosphate in the hexokinase reaction, like I just showed you. And we will later on talk about gluconeogenesis. That will be sort of the reverse of glycolysis, where pyruvate is converted to glucose 6-phosphate. And the pentose phosphate pathway gives us ribose 5-phosphate. And this is what we're talking about now, glucose 6-phosphate going to pyruvate. So the point of this animation is just to show the central role of glucose 6-phosphate in being connected to a lot of other pathways. So we can take glucose 6-phosphate to glucose, but not by hexokinase. So we'll talk about that later. All right, and pyruvate, when we're finished with that, will enter the citric acid cycle. And that's the subject for chapter 14. Pyruvate can be converted to amino acids, which we'll talk about in chapter 18, and pyruvate can be converted to lactate, and I'll get to that at the end of this lecture. All right, so let's go back to our slideshow. So we want to stop here and talk about how PFK1 is regulated because it is so important. So one of the things that we can see is that when the cell is energy poor, it means it's using up a lot of its ATP. And ADP is what is left when you phosphorylate, when you do a hydrolysis of that phosphoanhydrate bond. So ADP is going to act as an allosteric activator of PFK1. ADP concentration being high lets us know that the glycolytic pathway, which produces ATP, is needed. Phosphoenolpyruvate is a feedback inhibitor, so that will inhibit PFK1. It's not shown here, but ATP and citrate also inhibit PFK1. ATP indicates that the cell is energy rich and we don't need ATP, so that will serve as an allosteric inhibitor. So remember, the way that this works is that the allosteric activators, which I've highlighted in green here, they stimulate PFK1 by stabilizing the R form. That means that the enzyme has a high affinity for its substrate, whereas these allosteric inhibitors stabilize the T form, which has a low affinity for its substrate. So again, we want to be very conscious of the cellular energy status. High concentrations of ADP indicate that ATP is needed, whereas the opposite is true. High concentration of ATP means that ATP is not needed. I'm going to go to an animated figure here. Um, so this is the PFK1 reaction. So just to remind you, that involves fructose 6-phosphate, and it is phosphorylated to fructose 1,6-bisphosphate. So I'm going to scroll down a little bit here. So PFK1 is a tetramer, and it has R and T forms. And this is what the enzyme, if we take the enzyme, if we look at its activity, we measure that on the y-axis, and the fructose 6-phosphate, which is its substrate, on the x-axis, we get a curve, and we see that it's nearly hyperbolic, even though that we have an allosteric enzyme. But this is where we don't have a whole lot of ATP. So this is what we get. We get a sigmoidal shaped curve when we add ATP. So this is going to dramatically increase the inhibition. I don't think I'm going to be able to mark on this. But if we consider if this is the Vmax, and then we draw a line over and drop a line down to get the Km, we see we have a low Km here, but then when we add the ATP, the Km goes up. And remember, Km going up, a high Km means low affinity. So we have the PFK1 in its T form. Now, I would have drawn this curve in red because I think that's maybe a little bit misleading. So one of the things that this shows us is that if we have 0.5 millimolar fructose 6-phosphate, that's the substrate, that this is what the activity looks like if the ATP concentration is low. But when you add just 1 millimolar ATP, that you dramatically decrease the activity. So this is interesting, where you can still have the 1 millimolar ATP, but then you add a little bit of AMP, and AMP is more correctly called a D inhibitor, because this is the activity you got with ATP. And even if the ATP is still there, if you add a little bit of AMP, look how much you can increase the activity. So AMP relieves the inhibition caused by ATP. So we can call it an activator if we want. That's okay. But it's really more correct to call it a D inhibitor. All right, let's go back to our slideshow. So, so what I just showed you, this is a different graph from a different textbook, but it shows essentially the same thing, and you can see the changes in activity that you get whether you add whether you have very low ATP, you have high ATP, or you add this D inhibitor. So this picture right here shows the tetramer of PFK1, and the artist drew the fructose 6-phosphate in yellow. So we can't see all 4 of the binding sites. Again, this is a tetramer, so I can see one here and one here, and there are 2 other sites that aren't visible. But it's a tetramer, 4 subunits bind 4 molecules of substrate. And the ATP is sort of in this in this dark orange right here. So we've got fructose and we have ATP, because ATP is a substrate too, so that makes this a little bit complicated. But we need our fructose 6-phosphate plus the ATP, which is our substrate, and we need them to get together so that the ATP can phosphorylate the fructose 6-phosphate. So we have some other ATP binding sites over here. So these are the allosteric ATP binding sites. So these are the sites that will inhibit the enzyme and will convert it to its T form if the ATP concentration gets too high. So you do need some ATP because ATP is the substrate, but when the ATP concentration gets too high, the enzyme is converted from its R form to the T form. So I showed you this picture a while ago when we were talking about the T form and the R form of enzymes, and I wish I had an animation of this one, but I don't. So we'll just have to focus our attention on one form at a time. So in this pink color, this is the R form of the enzyme. And here's the fructose 6-phosphate. That's the substrate, and then this is the active site right here. So if we just look at the pink, notice that the conformation of the enzyme is such that there's an arginine that's in the active site of the enzyme, and that's going to form a nice ion pair between the enzyme and the substrate. And notice that what helps maintain the enzyme in this R form is ADP. So we're going to call this an ADP. This is an D inhibitor. So here's the ADP right here, shown in pink. So the ADP is binding not to the active site, but somewhere else, and it's stabilizing the R form. And in the R form, the enzyme has a high affinity for the substrate, as indicated by this ion pair. All right, now we're going to get rid of that now because I want to transition from the R form to the T form. So now pretend that the pink's not there and just look at the blue. So now we're going to have an inhibitor, and this isn't a natural inhibitor, but it's one that's very similar to phosphoenolpyruvate. So this is an inhibitor, and it is going to stabilize the T form. So the T form is shown here in blue. And what happens is there's a conformational change that occurs when you go from R to T. And what happens is that arginine right here swings over, so it's not in the active site anymore, it's sort of buried. And the other thing is that there was a buried glutamate before, but now the glutamate is in the active site. So now we're in the T form, and now the substrate's not going to be able to bind because the substrate is negatively charged, and if you put a negative charge in the active site, those charges are repulsive. So if we look at, we no longer have that arginine in the active site, and now we have a glutamate instead. The fructose 6-phosphate doesn't bind because it's repelled by that negative charge. So that's inhibition. What about stimulation? So we have a metabolite called fructose 2,6-bisphosphate. So it's not part of glycolysis. It's another metabolite. And since you're watching this now, you've already completed your top hat assignment. So fructose 2,6-bisphosphate is a potent activator of PFK1. And this is why we call it PFK1, because the way we get fructose 2,6-bisphosphate is there's another enzyme, another PFK. We'll call this PFK2. And that puts a phosphate on the number 2 position. So again, that's not part of glycolysis, but we do sort of siphon off a little bit of the fructose 6-phosphate. Another enzyme acts on it, and it makes this metabolite for us. So if you look at this diagram right here, you can see that this black curve shows no fructose 2,6-bisphosphate, but look at how much the Km decreases when you add the fructose 2,6-bisphosphate. So fructose 2,6-bisphosphate relieves that inhibition caused by ATP that I just showed you, and it stabilizes the R form that was in pink on that last slide. So the Km goes down. Km goes down means the enzyme has a high affinity for its substrate, and that is the R form. So what we should note is that fructose 2,6-bisphosphate is the most important regulator of PFK1. So when we get into more of this metabolism, I'm going to try and give you different strategies for figuring these things out and not memorizing. But you need to memorize this. So memorize this, that fructose 2,6-bisphosphate is an allosteric activator of PFK1, and it is the most important allosteric activator of PFK1. All right, moving on. We are on step 4. So here's our fructose 1,6-bisphosphate. We got through the first stage of glycolysis, and what happens next is we're going to do a cleavage right here, and we're going to cleave. Let's see if I get my pen back here. We're going to cleave the bond right here, and then we're going to, this half of the molecule, which is shown in red, is going to give us dihydroxyacetone phosphate. And then down here, which is shown in black, is glyceraldehyde 3-phosphate. So this enzyme is called aldolase because In the reverse direction, and this is reversible, you can tell by the arrows, in the reverse direction, it catalyzes an aldol condenation. All right, so the next thing that happens is the dihydroxyacetone phosphate is isomerized to glyceraldehyde 3-phosphate, and that's also reversible. So what I did is I drew a little schematic diagram of where we are now. So we took glucose and in 3 steps converted it to fructose 1,6-bisphosphate. And then we did the aldolase reaction, and we took this thing and we cleaved it into 2, and we got a dihydroxyacetone phosphate and a glyceraldehyde 3-phosphate. And then in this step that's shown on this slide right here, the dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate. So the net is we've started with glucose, and now we have 2 molecules of glyceraldehyde 3-phosphate. So here we are at the end of phase 1. So we took glucose and we phosphorylated it to glucose 6-phosphate. So that shows the phosphate right here. I'm not completely crazy about this diagram, but it's one of the better ones I have. It shows an ATP investment. And then we isomerize this to fructose 6-phosphate. And then we use another ATP to make the fructose 1,6-bisphosphate. And then we cleave the molecule in 2 to give us glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. And then the dihydroxyacetone phosphate gets converted to glyceraldehyde 3-phosphate. So now we have 1 glucose has been converted to 2 glyceraldehyde 3-phosphates. And we spent 2 ATPs, but we will get it back, I promise. Okay, moving on. What we really should do, which the textbooks do not do, is we should really multiply everything by 2 at this point. So see, I have 2 in front of everything because we have 1 glucose and 2 glyceraldehyde 3-phosphates. Okay, so there's a lot going on in this reaction. So this is step 6. We don't have to worry about regulating it, but this is the only redox reaction in glycolysis. So let's look at what's happening here. So if we look at, this is glyceraldehyde 3-phosphate, there's an aldehyde group right here at carbon number 1. See how we have 2 bonds to oxygen and 1 bond to hydrogen? And now we have 3 bonds to oxygen and no bonds to hydrogen. So that means the glyceraldehyde 3-phosphate has been oxidized to 1,3-bisphosphoglycerate. So it's lost protons and electrons. And you can't have an oxidation without a reduction, so the NAD+ picks up those protons and electrons to give us NADH and H+. So the enzyme here is glyceraldehyde 3-phosphate dehydrogenase. That's another thing to remember. So dehydrogenase, whenever you see an enzyme that's dehydrogenase, that's a redox reaction. And we can see that here, that the glyceraldehyde 3-phosphate was oxidized to 1,3-bisphosphoglycerate and the NAD+ was reduced to NADH and H+. Now, one of the things you should notice is this 1,3-bisphosphoglycerate was listed on that table that I gave you last time. It has a high phosphoryl group transfer potential. So let's go back to that table. It's right here. So you see how we have 1,3-bisphosphoglycerate. If we hydrolyze one of those phosphate groups, minus 49.4 kilojoules per mole is released, and that means that we can use some of that to couple ADP and inorganic phosphate, because the energy that's released when we hydrolyze the phosphate group on the 1-bisphosphoglycerate is more than the energy that it costs us to form a new phosphoanhydride bond here. So that's what's going to happen. And that happens in the next step. So look what happens here. See that phosphate? We're going to take that phosphate and we're going to take that phosphate and we're going to donate it to ADP to give us ATP, and then we end up with 3-phosphoglycerate. So this is phosphoglycerate kinase. You know, when there's kinase, you should go looking for the ATP. And of course, I had to multiply everything by 2 because remember, we have 2 molecules of 1,3-bisphosphoglycerate for every 1 mole of glucose that we started with. And that's important because I want to make it clear that we're getting 2 ATPs at this step. So the phosphoglycerate kinase catalyzes the transfer of a phosphate from the 1,3-bisphosphoglycerate to ADP to form ATP and 3-phosphoglycerate. So we're at the break-even point because remember we spent 2 ATPs and now we got 2 ATPs back. But the reaction is nearly at equilibrium and is not reversible. But we should know, if I asked you what type of ATP producing reaction is this, the answer is substrate level phosphorylation. So remember we said that there are 2 fancy terms for talking about how do you synthesize ATP. So this is one of them. Substrate level phosphorylation means that we're taking a phosphate from a substrate, in this case the substrate is 1,3-bisphosphoglycerate, and we're phosphorylating the ADP to make ATP. All right, going on. I'm going to multiply everything by 2 again. This is phosphoglycerate mutase, and you can see that this is, we could have called this an isomerase because the only thing that's happening here is there's a phosphate group is being moved from carbon number 3 to carbon number 2. We're going to need that in the next step, as you'll see. So we are carrying out an isomerization reaction. But when you say mutase, when you say mutase, think move. So we're moving the phosphate from the number 3 position to the number 2 position. So a mutase is a type of isomerase that moves a functional group from one part of the molecule to another. So again, not going to regulate the pathway here. This is a reversible reaction. Okay, so moving on to the next step, I'm going to multiply everything by 2. So I'm going to convert the 2-phosphoglycerate to phosphoenolpyruvate, and that reaction is also reversible. But what I want you to remember is that phosphoenolpyruvate is also on my list. It has a high phosphoryl group transfer potential. And here's that list again. And phosphoenolpyruvate is on top. So when we hydrolyze that phosphate group, we are going to release more free energy than it would cost us to make an ATP. So that phosphate is going to be transferred from the phosphoenolpyruvate to ADP to make ATP. And that's exactly what happens in the very last step. Let's multiply everything by 2. This is step 10. So we're going to take that phosphate group on phosphoenolpyruvate and we're going to donate it to ADP to form ATP. And now finally, we have generated 2 ATPs from the glucose. So we're at the end. This is the second ATP generating step. This is an irreversible and regulated step. We can see because the artists have helped us out here by just drawing the reaction with 1 arrow. So we should talk a little bit about how this is regulated. And one of the ways it's regulated is by fructose 1,6-bisphosphate and ATP. And I even have my color. So we have the inhibition is shown in red and the stimulation is shown in green. Oh, but we should also point out this is another example of substrate level phosphorylation, right? Because we made ATP and we donated a phosphate group from a substrate, phosphoenolpyruvate, to make ATP. So I did already sort of give this away, although you would have answered this on your top hat question. How does fructose 1,6-bisphosphate regulated pyruvate kinase? Well, you see that I didn't do this in green. But if you look here at the Km again, when you add the fructose 1,6-bisphosphate, again, notice that the Km goes down, and when the Km goes down, we have a higher affinity form, the R form. So fructose 1,6-bisphosphate binds to pyruvate kinase, not to the active site, but somewhere else, and stabilizes the R form. So the strategy here is to avoid a bottleneck. So if this is the last step, and if you have the high flux of metabolites coming through this pathway, you want to make sure that your last step doesn't hold you up. So fructose 1,6-bisphosphate is sort of a signal to the high flux of the pathway and indicates that the pathway the last step should not be a barrier to finishing the pathway. So I wanted to show you some animations here. Let me get rid of my pen tool for a second. And I'm not going to show you all of them, but I wanted to make you aware of them. These are posted on Sakai, and on the PowerPoint, the links are also live, so you can look at this on your own if you like. So let me put my notes up here first. So we're at the end. This is phase 2. So remember here, we should multiply everything here by 2, because remember when we started out with 1 glucose and we got 2 glyceraldehyde 3-phosphate, so everything here should be multiplied by 2. So the other thing that we should note, it just says 1 NADH here, but again, we have to multiply everything by 2. So we get 2 NADHs. So we invested 2 ATPs, we got 4 ATPs back, so the net yield is 2 ATPs. So let me show you these resources here that you can look at on your own. So this is just an animated figure right here. And what it does is it starts, it goes step by step. So we have glucose to glucose 6-phosphate. So that's the hexokinase. And so it gives you the reactants and the products. And it gives you the structure so you can see what they look like. So you can look at that on your own if you want. This is a guided exploration, so this is narrated. So if you go to this step right here, I'll just play the first part of it.
Glycolysis is the biochemical pathway through which a molecule of the 6-carbon
Turn on my
sugar,
volume here.
glucose, is converted to 2 molecules of the 3-carbon compound, pyruvate. In this process, free energy is sequestered in the form of 2 ATP molecules. The first reaction of glycolysis is catalyzed by the enzyme hexokinase, which mediates the transfer of ATP's gamma phosphoryl group to the number 6 carbon of glucose. The reaction occurs through the nucleophilic attack of glucose's 6-hydroxyl group on the gamma phosphorus atom, yielding glucose 6-phosphate and ADP. This is the first of 2 reactions in which the free energy of ATP hydrolysis is invested in the pathway.
Okay, so that was just the first step. So what that shows is that there's an animation of the reaction over here, and then it shows the reaction over here with the reactants and products. The other thing is that they have this little thing right here where they'll tell you when we've spent our ATPs, so we're 1 ATP in the hole now, but it'll show us when we get our net ATP. So so there are 10 pages here for each of the steps, and you can just hit next when you want to go on the next one. So I'll let you do that one on your own. So the last thing we need to talk about that's very important is that we cannot stop at glycolysis. When we go from glucose to pyruvate, we got the ATP that we needed to drive the various cellular reactions, but we also have this NADH, and we can't stop here because we have to do something with that NADH. And in fact, if we did go from glucose to pyruvate and that's all we did, you would essentially use up all the cellular NAD plus, and then convert it out to NADH, and then glycolysis would eventually stop if it didn't have any NAD plus. So the next step, whatever it is, and it could be different steps depending on what the needs of the cell are or what kind of organism you are, any of those steps, their strategy really is to regenerate the NAD plus. So I have a couple of these. Okay, so we can't stop at pyruvate because NAD plus must be regenerated for the glyceraldehyde 3-phosphate dehydrogenase reaction. So there's our NAD plus right here. So so again, if you have gone on to Top Hat, I've asked you a question about this last step here. But this is called homolactic fermentation. So I just want to make note of the vocabulary. So the word fermentation means that there's no oxygen involved. So this reaction occurs in us, in our muscles, if we're doing vigorous exercises when oxygen is lacking. So say like a sprint or something like that, something that's a very short duration. But bacteria do this in yogurt and cheese production. They convert glucose to lactate, and the lactic acid sort of curdles the milk. So what's happening here is that pyruvate, and again, look at this carbon right here that's shown in blue. This carbon right here is reduced because we have 2 bonds to oxygen right here, and we only have 1 bond to oxygen and 1 bond to hydrogen. So since the pyruvate is reduced to lactate, something else has to be oxidized, and that's our NADH. So this shows how NADH is regenerated to NAD+. And again, we can tell it's a redox reaction because it's a dehydrogenase. So what happens is glucose is converted to two pyruvates, and we get two NADHs. And then in that last step right here, when we go from pyruvate to lactate, that reaction uses the NADH and produces NAD plus, and that keeps glycolysis going. So we regenerate the NAD plus to keep glycolysis going. So if we took all 10 reactions of glycolysis plus that, say, that 11th reaction where pyruvate's converted to lactate, and if we crossed out all of the reactants and products on either side, we would get something that looks like this. So basically in homolactic fermentation, this is the sum of all 11 reactions where glucose is converted to lactate and we get two ATPs. And in the muscle, we can do this. The ATP generates, the NATP is, sorry, ATP is generated to power the muscle. So a lot of people have put forth the notion that lactate or protons, that if you have a lot of this, that makes your muscles hurt. But that's actually not true. A paper came out a while back that said that what happens is when your muscles get fatigued, they leak out calcium from the sarcoplasmic reticulum, and that weakens muscle contraction. So that's the answer to that question. It doesn't have to do with lactate or protons. So it's important to note that the energy yield is 2 ATPs per glucose. So again, when you go from glucose to pyruvate and pyruvate to lactate, overall that whole 11-step reaction gives us 2 ATPs. Now, this does not happen in our cells, but if you are a yeast, you can do this, and this is the basis of the beer and wine industry. So this is a two-step reaction where pyruvate, now here's another thing we can add to our vocabulary, decarboxylase. So if you take this carboxylate group and we release that as CO2, then we get acetaldehyde, and the next step, we can tell this is a redox reaction because it's dehydrogenase. Look at this carbon, 2 bonds to oxygen and 1 bond to hydrogen. And now look at that same carbon, 1 bond to oxygen and 1 bond to hydrogen. So that is the acetaldehyde has been reduced. So we need protons and electrons. So there's our NADH. And the NADH is then regenerated to form NAD+. So again, if this is our 10 reactions plus 11 and 12, say, if we took all 12 of those reactions and added them up, we'd find that we have glucose is converted to ethanol and CO2 and then ATP. So that's carried out by yeast in the production of alcoholic beverages. It also happens when you bake bread and the alcohol evaporates in the baking process. So again, 2 ATPs per glucose is what you get. So what happens here, to summarize, glucose goes to 2-pyruvate, 2 pyruvates go to 2-acetaldehyde, and then acetaldehyde goes to ethanol. And again, the NADH generated in glycolysis is used by the acetaldehyde and the ethanol reaction. And now we regenerate our NAD+, which is good. We have to regenerate our NAD+ for glycolysis to keep going. So the third choice is aerobic respiration. So again, let's note for vocabulary reasons that respiration means that oxygen is required. So the last 2 steps I told you, oxygen was not required. And you can see up here, if you go from glucose to lactate, there's no oxygen that's required. Now, you don't get a whole lot of free energy either. But if you take that pyruvate and if you completely oxidize all those carbons to CO2, you have to have oxygen in order to do that, and you get a whole lot more free energy when you do that. So when you go from pyruvate to CO2 and water, in this part of the pathway, the ATP yield is about 28 ATPs. There's actually a few more that are produced because you get the other 2 in substrate level phosphorylation, so that gives you 30. So remember that we only got 2 ATPs produced when glucose was incompletely oxidized to lactate. So we'll get 30 if you completely oxidize it. So you get a whole lot more ATP, but you got to have oxygen, and it's way slower. You do regenerate the NAD+, but you don't regenerate it until you get to electron transport, and that's the subject for chapter 15. So how does glycolysis fit in with animal metabolism? Okay, we started our lecture this way and we're going to end it here. So glycolysis is highly regulated. We need to coordinate glycolysis with all of these other pathways. So here, this is glycolysis right here, glucose to pyruvate. But glucose makes glycogen. If you're a plant, it makes starch and sucrose. If you need ribose phosphate, you go through the pentose phosphate pathway. If again uh, in animals, we would need extracellular matrix proteins. If you were a plant, you need cell walls. So it just depends on what you need as an organism, how you're going to use that glucose. So that is the end of our lecture today. I hope you enjoyed it and talk to you next time.